feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Welcome to the National University of Singapore Yong Loo School of Medicine Healthy Longevity webinar. Thank you for joining us. Dr. Austin Argentieri is our guest and will talk about a very novel topic, and that is the relation between the proteome, the genome, and its relation to the exposome. As always, we are starting with a short presentation, and this time by Shiva. She is a research assistant and a PhD student talking about high-fat diet interventions in aging mice.
Thank you for the introduction. As mentioned, my name is Shiva and I'm a research assistant in Prof Brian Kennedy's lab. And for my presentation today, I will be talking about an upcoming mouse intervention study with high-fat diet that we will be conducting with a promising compound. The initial longitudinal mouse study with the low dose of this compound showed us positive results in female mice, such as reduction of frailty, an overall reduction of chronic inflammation, and a reduction of aortic pulse wave velocity, which is an indicator of aortic stiffening. So with a high-fat diet study, we hope to further understand the effects of a diet that more closely mimics human population diets and the impact the low dose of the compound would have on that. It would also allow us to better observe potential aortic lesions in C57 wild-type mice, as well as the impact of inflammation on aging that has been induced by a high-fat diet. High-fat diets can consist of different percentages of fat content and can also include high sucrose or fructose levels. C57 wild-type mice on a high-fat diet have shown to have weight gain, insulin insensitivity, and high levels of lipids in their blood, also known as dyslipidemia. With aging, this is further exacerbated. Based on the effects of the compound on a healthy diet, we would expect to see overall reduced high-fat diet-induced inflammation that would ultimately result in a similar reduction of aortic stiffening as previously observed. This high-fat diet intervention will take place with mice from 18 to 22 months old, either on a high-fat diet alone or on both a treatment and high-fat diet. The treatments can be divided into two different types, one of which takes a look at the potential preventative effect of the compound by starting the mice on a diet of the compound for a month before the start of the high-fat diet. The other treatment will have the mice on high-fat diet and compound together for 4 months, which will allow us to observe its more therapeutic effects. As the use of high-fat diet will accelerate aging and reduce the overall lifespan of the mice, the treatment can be conducted over a shorter period of time compared to our usual longitudinal mouse studies. During this study, we will be conducting a frailty test every 2 months based on the 31-point frailty index published by Whitehead. This frailty index will allow us to measure any health deficits that are observed with aging. The DEXA machine will also be used to measure body composition of each mouse before and after respective treatments. With this, we are able to measure bone mineral density and content, fat tissue and lean tissue, etc. By measuring for these, we will be able to understand the effect of the treatments on retaining muscle versus fat, since weight alone might not account for the differences in the percentages of them. In addition to the general health and frailty of the mouse, this project also focuses on the cardiovascular system, specifically the aorta. On standard chow, C57 mice rarely show any aortic lesions or have very minor changes in the aorta. With the high-fat diet, we are more likely to observe aortic lesions, and so with the treatment, we will be more aware of any effects on the aorta. Ultrasounds allow us to look at the structure of the aorta, such as the diameter, and measure aortic pulse rate velocity. We will also be conducting behavioral tests, such as grip strength, rotor rod, and treadmill, that will test for motor and muscle function. In addition, tests such as Barnes maze and novel object recognition that test spatial learning and memory will also be conducted. At the endpoint, both organs and blood will be collected, with which further analyses will be conducted. These will provide us with a comprehensive list of information about various factors in the blood and how they might have changed or been maintained with the respective treatment. In addition, blood plasma will be used to conduct Luminex that will tell us more about the inflammatory state of the mice on high-fat diet and if this was mitigated with the treatment. In summary, with this high-fat diet study, we hope to further understand the effects of a diet that more closely mimics human population diets and the impact the compound would have on overall frailty, inflammation, and cardiovascular health. That's the end of my presentation. Thank you for your attention.
Thank you, Shiva, for this very nice presentation. I would love to see these studies also in humans. But now to our guest for today. We are talking about the proteins and the exposome. Dr. Austin Argentieri is a research fellow at the Massachusetts General Hospital with an academic appointment at the Harvard Medical School, but also at the Broad Institute. His research is focusing on large-scale analysis to understand the genetic, biological, and environmental determinants of human aging and age-related diseases. Austin, it's our pleasure to have you here. I think it's a very, very novel topic, and I would love to learn with my genotype if I should have exposure to the exposome in Singapore. Maybe I will learn that.
Great. Thanks for having me. Thanks, everyone, for joining and for watching. Today, I'm going to talk a little bit about our work on developing aging biomarkers, mostly through using proteomic information. But I'm really going to use that as an opportunity to discuss kind of more broadly our approaches and the principles and the pillars that we are trying to follow in developing aging markers, specifically as tools that we can use in the context of preventative medicine. So these are most of the aging hallmarks that we know exist on a biological level that are conserved across species. But the really fascinating thing about these aging hallmarks and the strategic thing about these aging hallmarks is that they seem to be shared across many different age-related diseases. And so it's a common underlying biology across these different diseases. And so the real opportunity when we're thinking about studying aging for the context of preventative medicine is that it might afford us this opportunity to develop tools and biomarkers that can help us predict future risk of many different diseases simultaneously, which might allow us to develop then tools that can allow for the prevention of multiple different age-related chronic diseases simultaneously. Among the different hallmarks of aging that you can see here, one of the major hallmarks is loss of proteostasis. And so we had this question for ourselves for our own research: can we use blood proteomics information, which is readily available, to quantify signatures of aging that are then accurate tools for the detection of risk for multiple different chronic diseases simultaneously? And so let's put that into global population health context for a second here. What I'm showing on this slide are the 10 leading causes of death globally, and 8 out of these 10 diseases are age-related chronic diseases. And together, just these 8 diseases alone accounted for nearly 26 million global deaths in 2021. So these are major killers here, but as I mentioned in the previous slide, the real strategic opportunity here is that these major causes of death share a common underlying aging biology. So again, if we can develop tools that allow us to detect and measure that aging biology, they might be really strategic tools for us to use within the context of preventative medicine. So with that said, I'm going to go through here some of the different principles that we've been trying to follow in developing our own aging biomarkers, again, for this context of preventative medicine. And the first and maybe one of the most important pillars or principles that we've been trying to follow with this is that the aging biomarkers that we develop, they need to work for everybody. These are tools that need to work for everybody. And there's a scientific rationale for this. We need reproducibility, we need reliability in the tools and the measures that we develop, but there's also an ethical imperative for this. We need to be able to make sure that the tools and the aging biomarkers we develop work for everybody so that we're not sort of inadvertently contributing to growing health inequalities locally or globally. That's something we want to avoid at all costs. And doing these things is what's really going to allow us to make good on the promise of really using these aging biomarkers in a sort of personalized medicine context. So I'll introduce here some of the work we've been doing, develop, like I said, a proteomic aging biomarker. This is from a paper we published last year in Nature Medicine, where within the UK Biobank, which is a sort of population biobank in the UK, we developed a proteomic age clock using about 3,000 different plasma proteins they have available there. But importantly, we incorporated into our approach from the very beginning, making sure that we develop and test our models, not just that they work in the UK participants in which we sort of trained everything, but that they work in globally independent and external data sets as well. So we also validated this proteomic age clock in 2 different biobanks, globally, one in Finland called FinGen, another one in China called the China Kadoorie Biobank. But these populations were very different than the UK population in which we trained all of our proteomic age clock models. So there are very different age profiles from one another, which you can see in the figure at the bottom of this slide here. There are also, of course, different genetic ancestries, different geographic regions of residence. There are also very different morbidity profiles. The Finnish participants were largely healthy and free of any major morbidities. The China population, on the other hand, half of them had ischemic heart disease, the other half were random population controls. And the idea here is to be able to develop models that work across broad swaths of the population so we can find what's common in the biology across everyone, and we can develop tools that are much more generalizable to everybody in the context of clinical care. So this shows the performance of the proteomic aging clock we developed. You can see across these 3 different panels. This shows on the x-axis the chronological age of participants, and on the y-axis, it's the protein predicted age of each participant. And you can see the correlation between those is very consistent across both the UK population that we started with on the left in blue, the China population in the middle in red, and then the Finnish population on the right in yellow. And the primary trait or the phenotype that we pull out from this is what we call the proteomic age gap. It's the difference, in our case, in years between protein predicted age and chronological age or calendar age. And it's this trait, this proteomic age gap, is what we really pull out from this and we use as our sort of predictive biomarker for all the different tests that we do sort of downstream, which I'll talk about in a couple of slides. But the thing that we found in this study is that when we sort of developed this in a way that was generalizable to those populations, that the distribution of this trait was very stable across different ways that we can cut up both within these cohorts and across them. So you can see here on the left, for example, the distribution of the proteomic age gap across the sexes in all of the cohorts that we studied. In the center here, the distribution of the proteomic age gap across different self-report race ethnicities in the UK. And then on the right, the distribution across different geographic areas of residence in China, which is one of the major sort of population differences that contributes a lot to differences in disease risk and things like that in China. And again, here across all of these different ways of cutting up these populations, you can see a sort of remarkable consistency in the distribution of this proteomic age gap phenotype. And we see the same thing even when we look in the UK population across different genetic ancestries here. So you can see on the left, despite the fact that, especially Europeans compared to the other genetic ancestries have a somewhat different age distribution or the distribution of the calendar age of participants. If you look on the right, still we see a pretty stable distribution of this proteomic age gap phenotype across even just these genetic ancestries within the UK population that we studied alone. Okay, so the next principle we've really been trying to follow is reducing complexity, right? The simpler we can make these aging biomarkers, the better. And there's a pretty straightforward reason why. If we can reduce complexity in our models, we can reduce the complexity of the biological assay we need to capture and measure that information, which is going to translate to reduced cost. And that's really what's going to open up the accessibility of these sort of aging biomarkers for preventative medicine. That's going to allow not just those who can afford it, but really broadly to everybody to be able to use. We want cheap and easy. So in the context of our work, we spent a lot of time trying to see if we can find much more smaller models that capture the same performance. One of the things that we find here is that you don't need thousands of proteins to get an accurate proteomic age clock. So in our case, what we found is that you can go from 3,000 proteins all the way down to just 200 proteins with virtually no drop in accuracy and develop a proteomic age clock. But perhaps even more interestingly, we found that you can go all the way down to just 20 proteins and still capture about 95% of the overall accuracy of these kinds of models. So a great takeaway from us here was that you can do a lot of work with just 20 proteins in predicting age and having an accurate aging biomarker. And as I'll show in some of the subsequent slides, even just these 20 proteins are still going to be highly predictive of a number of different age-related diseases. So that was very important for us to be able to reduce this dimensionality here. Okay, so that kind of segues into the next principle, which is if we're thinking about developing tools for the context of preventative medicine, that means that these aging biomarkers we're developing should be capturing future risk of disease or frailty before the onset. We want to be able to understand someone's future trajectory, someone's future risk of disease, so that we can intervene before it happens and we can change their future risk trajectory. So in the context of the research that we carried out, what we found was that the proteomic age gap phenotype that we measured, whether from 200 proteins or 20 proteins, is associated with future risk of mortality and 18 major age-related chronic diseases. And here in this slide, I'm just showing the results for the non-cancer associations, but there are a few cancers that it's also associated with here. And what this slide shows is that these proteomic aging clocks are associated with all of these diseases simultaneously. And this is showing future risk of the disease. So this is incident disease, those who don't have the disease at baseline and then who goes on to develop it in 10 to 15 years of follow-up afterwards. And this really seems to demonstrate that we are capturing something here, and that's this more fundamental aging biology, because it's really associated broadly across all of these aging-related diseases, which are many of the sort of major killers that I showed in the second slide of this presentation. Here in this plot, I'm showing the hazard ratio of the association between the proteomic age gap, whether it's 200 proteins or just 20 proteins, in a Cox model with each of these incident outcomes. And this is the hazard ratio per 1 year of proteomic age gap. But the distribution of that can be, plus or minus 10 years of proteomic aging, or sort of difference in protein predicted age versus chronological age. So to put that in broader context here, comparing those who are in the highest distribution of this proteomic age gap versus those that are in the lowest, it's a 6 to 7 times increased risk of Alzheimer's disease. But this tool that we developed, this proteomic age clock that we developed, is also a really powerful tool for stratifying participants in terms of their future disease risk. And that's what we're showing in some of these figures here, where for each of these different diseases and mortality that I'm showing in these different plots, you can see the cumulative incidence of all of those over the 10 to 15 years of future follow-up. So you see that according to those that are in the top 10% of the proteomic age gap in blue, those that are in the median 10% of the distribution of that trait in orange, and those that are in the bottom 10% of that distribution in green. And the takeaway here really is that no matter what the age of the participant is at baseline in the 10 to 15 years in the future, this aging clock is able to really strongly capture or stratify people into different risk trajectories based on just readout of a few hundred different proteins. And not only does it tell us, are we able to use this to put people into the buckets of who are very high risk of these future diseases, but it also allows us to differentiate who are those who are very low risk. So for an example, if you take a look at the all-cause dementia and Alzheimer's disease down there, even among participants who are already 65 years old at baseline, if they're in the bottom 10% of this distribution of this proteomic aging gap, then across 10 to 15 years of future follow-up, less than 1% of those who are in that bottom 10% of the distribution will go on to develop all-cause dementia or Alzheimer's disease. And so that reaches participants all the way up to the age of 80 years old, give or take. So it's a really strong, these are really powerful ways for us to really stratify people into what their different health trajectories look like in the future, given that they don't have a disease at the time when we measure the blood. And I can show this for the sort of rest of the sort of non-cancer diseases we looked at in the UK, sort of similar stratification across these. But this also works in the China cohort as well. And again, we didn't train or develop any of this model in the China cohort, but we were able to bring it into the China participants. And it sort of shows a very similar effect or ability to stratify risk across all these participants here. And here we're limited a bit more in sample size in the China population, so we can't show quite as many diseases, but I'm showing here all the diseases where we had, I think, at least 10 incident cases across follow-up in the China cohort. And again, you see a similar ability as to stratify risk in this cohort as well. We didn't do this for the Finnish cohort because, like I said at the outset of this talk, those were mostly healthy participants with no morbidity. So we didn't have the opportunity to look at how it stratifies disease risk in Finland. But, you know, the fact that it associates still with age strongly, and it can capture age very strongly, even among those who don't have any disease, is important as well. We want to be able to deploy these models in people who don't have disease, and we can look in the future. So the next principle that we've been thinking about in sort of developing and refining these aging biomarkers that we've been developing is thinking about leveraging human genetic information to improve the confidence in what we're measuring and how it's connected to different phenotypes of interest. There's a lot of evidence that demonstrates that incorporating genetic information and understanding the genetics of your trait of interest, for example, can really improve your understanding of what your rate of success might be, if, for example, in the context of a clinical trial. But it also, using the genetics, allows us to establish different shared biology between the biomarkers we're developing and different important aging traits that we might be interested in. So we have a lot of ongoing work on this in our lab, but a lot of it's unpublished, so I won't necessarily show any of it here, but I will sort of discuss a little bit the rationale and the background for why we've been doing a lot of this. So in the context of the drug development and the therapeutic development world, it's now been well demonstrated in a very recent paper that I'm showing here, but earlier papers before that as well, that drug mechanisms with genetic support are nearly 3 times more likely to successfully pass through clinical trials and receive regulatory approval. By genetic support, I mean that there's a genetic association between the gene corresponding to the drug target and a trait that's equal to or similar to the indication for your clinical trial. And what I would argue here is that we need to be thinking similarly about making use of this strategy when we're developing biomarkers of aging, particularly those that we want to use in clinical settings, that we might want to use as surrogate endpoints in clinical trials, all of these things. So understanding and carrying out analyses where we can look at the genetic associations with these different aging biomarkers and then connect those genes or those genetic determinants of the aging biomarkers to age-related diseases we care about. We can use that to start to demonstrate causation using Mendelian randomization. We can use different techniques like genetic correlation and co-localization to understand the shared biology between our aging biomarkers and different aging traits and diseases that we care about. So this is a toolbox that's gone to very commonly for the world, again, of drug development and therapeutic development. And I would argue we should be thinking very similarly about this problem in the context of developing biomarkers of aging for either clinical trials or preventative medicine more broadly. So that then brings me to our sort of last principle that we've been thinking about that's very important, which is that aging biomarkers should be modifiable, and we should be able to identify what are the different inputs, what are the different possible interventions or treatments that allow us to change and modulate those aging biomarkers. And that's because these types of tests and tools are probably most useful in the context of testing early and often, right? We want to be able to have an early test where we can understand someone's future disease trajectory And I've shown in some of our previous slides that we can do that in a window of 10 to 15 years in the future. But the idea is we want to test early. We want to understand someone's future risk trajectory. Then we want to be able to do something about it. We want to be able to administer interventions. We want to be able to administer therapeutics, and then use these biomarkers to monitor in real time the sort of biological success of those interventions and allow us to course correct and refine from there. So these need to be modifiable. We need to understand and identify the levers that allow us to change these future disease risk trajectories. And that's what's going to unlock the preventative health opportunity in all of this. So some of the work that we've been carrying out trying to identify these levers comes from a paper we published just a couple of weeks ago in Nature Medicine, where we carried out what we call an exposome-wide analysis of aging and mortality. We did this in the UK Biobank again, but now in the full participant population, which is almost 500,000 participants across the UK. And what we did is, we defined the exposome really broadly as a couple hundred different measures across early life and childhood that capture different exposures in someone's environment, whether it's air pollution, smoking, physical activity, what things you were exposed to when you were a child, psychological well-being, all these kinds of things. And we first started by carrying out an exposome-wide association study, which is kind of akin to a genome-wide association study, looking at the association of each of these different exposures with mortality. And then as a secondary step, looking at the association between each of these exposures and the proteomic age clock that we developed. And the idea here was that we wanted to find exposures that have a consistent association between both future mortality and proteomic aging, and that will help us identify maybe some more reliable subset of exposures that seem to be important for aging because they both are influencing or associated with ultimate lifespan, but also the intermediate aging biology as well. And we had a number of other different steps that we took to try to sort of weed out some of the residual confounding in these observational exposures that allow us to get to a more reliable subset of exposures that might be sort of important drivers for aging and mortality. So once we do that and we're able to get to that subset, then this really unlocks something pretty powerful for us. So on the x-axis here, you can see a list of 25 different independent exposures that we identified within the context of these 500,000 UK participants that seem to be reliable, sort of reproducible exposures related to mortality and aging. And the nice thing here is that once we are able to identify that subset, they show beautiful patterns of sort of consistent associations across all the steps in the causal pathway, going from initial exposure to changes in early biology, to then changes in future disease risk and changes in mortality. And being able to map this across all those stages of the causal pathway really allows us to improve our confidence that these are going to be important levers because they're going to be associated with all these different steps. So what you're looking at in this figure here is, again, all of these different exposures and their associations with, in the bottom panel here, future risk, so incident risk of all of the different age-related chronic diseases that are either major causes of death in the cohort or highly prevalent in aging populations. The associations with our proteomic age clock here, the association with different clinical risk, disease risk factors like hypertension, obesity, and dyslipidemia. And then on the top here, it's showing the association between each of these exposures and a number of different biological mechanisms that are either markers of organ function or some aging-related ones like telomere length, things like that. And what we see here is sort of a remarkable consistency and significant associations between each exposure and all of these combined. So if we look on the right, for example, you have exposures like education, physical activity, income, living with a partner, which is kind of a social support. Each of these are associated with future risk of many different age-related chronic diseases with lower or sort of decelerated proteomic aging, with decreased hypertension, obesity, dyslipidemia, sort of lower levels of detrimental biological mechanisms and higher levels of positive or protective biological mechanisms. So a real consistency across these, which gives us confidence that these exposures are really drivers of this whole process and drivers of this kind of more fundamental aging-related multimorbidity that we've been talking about throughout this talk. There are, of course, then the detrimental exposures, things like smoking, sleeping enough, sleeping too much, things like this that are in here, which show the exact opposite pattern of associations across all these different layers and stages here. Um, and so the takeaway from this is that some of these exposures we can identify in this way. None of these are necessarily new in the context of epidemiology, but this type of exposome-wide approach, bringing in all of the different other layers of the biology and the disease risk, allows us to identify what are the really high priority exposures that if we can move the needle on these, it's going to move the needle on all of these downstream things in multimorbidity and age-related disease. And it's going to, help us make good on this promise of sort of like, look at one area and then it's going to have this downstream impact on all these different areas because we're looking at things that influence aging. Right. And the significant exposures here are really basic and fundamental. Education's different aspects of socioeconomic status, sleep, smoking, physical activity, social support, psychological well-being. If we can move the needle on these, it's going to have a tremendous downstream public health impact on all and many of these different major killers simultaneously. And sort of just as a final slide to talk about this work, the sort of added benefit of being able to do this kind of exposome-wide types of analysis is then it allows us to sort of for the first time in the context of these big biobanks to add up what's the total contribution of the environment across all these exposures, for mortality and for different diseases that we care about. And then we can use standard tools like polygenic risk scores, for example, to estimate the total contribution of at least common genetic variants to these outcomes. And we can start to do sort of a comparison. How much is the environment versus genetics contributing to explaining risk for mortality in these different diseases? So you can see here kind of right in the middle, the results for all-cause mortality. And what we end up finding is that sure enough, the contribution of the environment in explaining risk for mortality is much greater than the contribution of polygenic risk. And the way we looked at genetics, the genetic contributions in the case of mortality is we took actually 22 different polygenic risk scores for all of the different diseases or almost all the different diseases that you can see here. So all the major disease-related causes of death in the UK at least. And so what we end up finding is that compared to a model with just age and sex, the exposome explains an additional 17% of mortality variation in this cohort, but compared to age and sex, polygenic risk explains less than 2%. So the contribution of the environment to explaining mortality is nearly 10 times as great as the contribution of polygenic risk. And together, all of those combined are, account for 70-something% of the overall mortality risk in this population. But what we can also show is we can do this across all the different diseases that we study as well. And you can see across diseases, it's kind of a mixture. Unsurprisingly, for some lung-related diseases like emphysema, COPD, lung cancer, it's really exposome that's dominating our ability to explain risk, but also for some diseases like chronic liver disease, which you can see all the way on the left of the slide here. And then for other diseases, it's really the genetics that's dominating, or it's age and sex that's just dominating across everything. But for dementia and Alzheimer's disease, for example, you can really see that the genetics has a more important contribution, at least in terms of proportion, than the environment. So, we like to think about a lot of this sort of exposome-related work as achieving two things for us. One, it allows us to do these comprehensive views and to understand, how much of variation in these different age-related outcomes is explained by environment versus genetics. It allows us to get at that fundamental question. But as I mentioned in our previous slide, perhaps one of the most powerful things is that it really allows us to identify what are the most high priority exposures that are going to give us the most bang for our buck, so to speak, in really moving the needle on this whole downstream landscape of aging-related disease, multimorbidity, and which then ultimately, obviously, influences mortality risk. So with that, I'll just say thank you to all of our collaborators, and I will thank all of you for joining and listening.
Thank you, Austin, for this wonderful presentation. I learned a lot, and I will absolutely watch the space about the proteomics. And I think you as listeners, hopefully you as excited as I am to now really implement the biological age measures for individuals and maybe for ourselves. Leave any comments you have in the chat function. I have two announcements to make. If you are a junior in the health longevity field and want to expose yourself for 12 days to an innovative and challenging exposome, to learn about geroscience, to learn about precision geromedicine, make your network building a strong basis for healthy longevity, then I think the Healthy Longevity Talent Incubator might be for you. It's happening from 30th of June to the 11th of July at University Town in NUS. So applications are very welcome. Then, if you would like to participate in one of our studies or you have a neighbor or a friend, we have Life Codes. Life Codes is a randomized control trial that investigates evidence-based lifestyle interventions, which can possibly delay the cognitive decline in at-risk older Singaporeans aged 60 to 75. So please help us to recruit, knock on the doors of your neighbors, and invite them if you are living in Singapore. The next Healthy Longevity webinar is at the 10th of April. Kevin Paris, a junior group leader from the Charité, beautiful Berlin, at the Universitätsmedizinzentrum will actually present. And I will leave you with a final video. And I can tell you, aging begins at the molecular level. Take care.
Is science going to actually cure death? The idea of why we age, or can you reverse aging or be immortal, has actually been a topic of interest for centuries. I think a lot of people don't realize how much power we actually have over our aging process. Most people think of age or aging in terms of chronological time. So we all know how many years we've been alive, and we usually measure our aging in terms of months, days, years since we were born. We almost give it a magic number. So you're old when you're 65, or you're old when you can see it in the mirror. And we put a lot of emphasis and importance on this measure, but actually, this isn't the number that counts. Scientists think aging is starting even before birth, and it's really this continuous process that's happening over our entire lives. The most obvious thing that people associate with aging is changes in their skin, changes in body composition, graying of hair. We experience functional changes, our ability to walk, our balance, vision, hearing, and also changes in our risk of developing different diseases. But in reality, these aren't where aging is actually starting. These are what we would call the emergence or the manifestations of aging. We really think aging is starting at the molecular and cellular level. And once that reaches a given threshold, that's when we actually can start seeing this in the mirror in our everyday lives. I'm Dr. Morgan Levine. I study the science of aging, and my book is called True Age.
Like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my I